I'll make an attempt on all three of those. The first, so human data and genetic data, in a sense, that certainly in Carolyn's work and in our work, it's there. It's not explicit, but it's there in the sense that we're using the associations that have come out from the whole genome scans or the single gene, single disease associations as the underlying biological facts that hitting this gene or messing with this gene is associated with this disease. So since we don't have new data, but we're using that data to inform what we learn from the analyses we're doing. So they're almost in a Bayesian way, they're prior hypotheses about what we should expect to see when we look at interactions between a particular assay on a gene and a chemical. On the release of the raw data, we've discussed this with Chris in the past. With certainly all of our data, that turns out to be a complicated workflow question. It's not theoretical at all, and we're actually moving towards releasing all of that data so Chris and whoever else can play with that. So you'll actually have all the nasty, noisy dose response data, and people can come to their own conclusions from that. Let's see, what was the third question?
Before we go into the growth pains and sustainability of the effort, I think we can take some time and discuss the human relevance and also raw data and also issues of getting lost in databases and how to remedy that.
So we do represent the genetic associations with diseases by virtue of our integration with OMIM and additional curated gene-disease relationships that we find that are not necessarily represented in OMIM, which is not updated super frequently. And in reality, there really aren't that many explicit gene-disease relationships. So I think that's not going to be a huge data set to start with. I think what is more problematic is what are the genes that are being perturbed by exposures, obviously, and how does that contribute to disease? And in terms of the balance, I think, and raw data. So raw data is just, I think it would be very difficult for a single database to be everything to everybody. And I think there are some groups that are really well suited due to the size and funding that they have to do a better job at storing the raw data than we are, for instance. But I think what's really important and what I was thinking about a lot listening to these talks is the importance of both, because I think even though we don't include the raw data, what we're including is our data where the validation has happened in most cases. And I think that's really nice to set on top of some of the high throughput data so that you can get some perspective when you're looking at these large data sets. What is the state of knowledge? These aren't just data points given some specific reflection of what the experiment was done at that moment. But having a combination of both, and I think maybe we could, this might be an opportunity for some of our groups to get together and think about what a meaningful way of integrating our resources might be.
So before your next question, I have one more perspective on the human side of it and how important, the kind of models that we're trying to develop to fill in where you just can't get the human data. And a project I got involved with that I didn't talk about today was generating an in vitro micronucleus assay using bone marrow derived progenitor cells to do erythropoiesis. And it arose out of conversations with folks at DuPont and at Amgen about limitations of existing micronucleus assays where you have CHO cells or some other in vitro cell and you get a positive result. It's not clear what it means. And together with Harvey Lodish, a stem cell biologist, and Leona Sampson, who studies DNA damage and repair and head of our CEHS, we had a student look at the role of DNA damage and repair enzymes on this in a developmentally regulated way. And it's really clear you have very intricate balances between damage and repair at different developmental stages. And so as we go in, these are going to be different in humans and mice, and there are now companies like Stem Cells in Vancouver who is getting huge number of requests to do these kinds of assays for clients who are drug companies, but it comes back to the quality, super high throughput screen. You're not going to do that with human bone marrow progenitor cells, but you get a lot of information about how complicated systems interact that's perhaps more relevant for the human system. So there's some parameter space there that I think we're on the verge of building more robust technologies. They're going to be more expensive, but they're going to be way higher information content relevant to humans to throw in to these kind of databases. So I'll just make a plea that we still are interested in mechanistic studies that would explore in a detailed way, lower throughput, some of these things.
Before we move on, Robert, let me ask you a question. Maybe you can also add on some of these model organism data. What is your raw data level or format? And you did talk briefly about the numerical values or types of scores you're trying to develop. But when you publish papers or release data, I mean, do you, from academic, collecting data in academia, some on the grants, other on contracts, what do you do with the model organism data sets as well?
Yeah, that's a good question. We do have a knowledge base. It's not a database. It's really a knowledge base, and we have access to all of the raw data. It's in a format that's probably not real amenable to what Carolyn's dealing with right now. But we report absolute raw data. You can drill down and look at actual image-based, in fact, and it's challenging to get our endpoints to correlate with some of the other endpoints, other systems. And I think that's the barrier right now, trying to be able to talk the same language, because we do have similar compounds, similar dose metrics. We just have different responses. So how do we make sense of it?
And before we move on, I just wanted to probably add that as the committee is considering future topics for workshops, I'm sure that both the issues of inter-individual variability and bringing genetics, and also some other topics that we're discussing here, such as stem cell and other types of technologies that can be used, will be covered in the next couple of years. So let's move on to the next question. And Mark?
I'm Mark Johnson from the Army Center for Health Promotion and Preventive Medicine. One source of confusion in my mind is the use of the word screening with toxicity testing. And so what I'm hearing is the word screening in this context is very specific, isn't it? And I was wondering if you would expand on that and how we can better find out what the holes are when we talk about improving our way to screen chemicals for toxicity.
You mean screening, collecting data, and toxicity testing, really trying to understand the mechanism, right?
Well, when you have a new compound, you know absolutely nothing. Where do you begin?
I think it's going to begin at least with a multiple set of tests that can be rapidly done. And I would argue that a cell-based screen coupled with, say, a whole animal screen is a really nice place to start, because the speed at which you can get it and the amount of compound that you need to do those two types of tests are minuscule. And we might be able to triage very quickly identify toxic responses very rapidly with, say, your unknown compound within a day. So I think we should. What seems to strike me is that we continue to have a lot of coordination problems. There's lots of parallel activities going on, and it seems to be these workshops where we all recognize it and then we go off and continue to work in parallel.
So I think it's hard to talk about screening without talking about the objective. And so I'll give at least the EPA our take on this is that we're screening to be able to prioritize chemicals. Okay, so these in vitro screens are not going to answer the question, is this a carcinogen? Is it an endocrine disruptor? It will give you enough evidence that you'd say, okay, out of the 10,000 compounds we look at, these are the X number that we want to go and dig deeper into. And there's actually a fundamental argument we're having about what level of evidence do you have to have at the end of that screening to carry forward. And I'll just say the two lines of thought. One is all of our current in vivo data, for better or for worse, is rodent and rabbit and so on. And so you come up with some model, like the one I showed, where you have some set of assays where you run, and the outcome of those assays, in vitro assays, are correlated with some sort of a rodent endpoint. And so they're trying to mimic the current bioassay that you use to regulate on. Alternatively, and so that's sort of a correlated fact that you may not understand the biology. Alternatively, you can simply say, okay, there are certain targets, so the androgen receptor, the estrogen receptor, the various sort of kinases, where we understand from drug development or wherever that if you actually do hit those targets and do something strong to those targets, that could have an effect. Okay, you don't have any correlative reason to believe, to understand how you map from an effect there onto an effect in a human, but it's just sort of an accepted fact. And so that's we don't quite know, we haven't come to a conclusion today what level of evidence you need from these screens before you would go and take some other actions.
Richard Beeger, NCTR, FDA. Most of the talkers today said that there was problems with metabolism. And I agree that metabolism is a problem because a lot of toxicity, a necessary ingredient is certain metabolites. Is there a way that quality control can be put into some of these studies that look at, so we have an idea how to compare in terms of metabolism? I'd be interested to see what they say.
So we have taken that on in other groups using our model. You actually can do bioactivation steps. I was talking to Linda about that earlier. You can actually, in principle, bioactivate your entire testing library and have like a pre and a post metabolism, and then compare the two. So we're trying to find ways to do that in more efficient ways, but it is an issue, and we have some great examples of compounds that should be highly toxic and they are not in our system, and we metabolically activate it, and sure enough we can get it. But again, that's because we knew. We're in that unknown world.
There are technologies coming out to marry metabolic activation with cell cultures. There's a company called Solidus that is introducing these really high density chips where they can do an activation, put the drug on, do activation step with liver cells, and then contact it with another cell type like a mammary epithelial cell. And John Dordick is the lead person on that if you want to look up his paper. So there are things coming online, not only to do that with like S9 fractions, which you can get from any company, but to actually use cells for that. And again, in high throughput screening, so you're not going to get as much functionality out.
Richard, can you add maybe something on ToxCast? What are the controls of at least what's in the well versus what we thought was put in the well, and what are the thoughts about the metabolites versus the parent compound in screening?
That's a leading question. So what we're doing is we're screening a whole series of pesticides against hundreds of assays, and we actually ran the Solidus assay. That was one of the assay panels, and so it's actually telling us something about which chemicals need to be activated, bioactivated to actually cause their toxicity. We certainly don't have the answer from that, but just say that it's an important, it's an important issue that we don't understand the answer to yet.
There is also data coming out from these HTS assays where there is a parent and a major metabolites that have been screened. So there is some data that is possible to then interrogate for that potential question.
Yeah, so we actually did, where we know the major metabolite, in a handful of cases, we did both the parent and the metabolite, and you get, in most cases, you get the expected answer that the active metabolite is more toxic or it's more interacting with a particular interesting target.
So I would like to welcome 2 more questions from the committee, but also would like to remind everyone that we'll have all the speakers today in the larger panel discussion, so there'll be additional opportunity to discuss some of the challenges that we will not bring out today in this very short kind of follow-up panel. George?
Do you want me to wait, or? Okay. Well, I wanted to make 2 points. One is about the amount of information that ought to be included in databases. As we started to get into microarray experiments, but I think that this also counts for all the high throughput work and as well, is we expanded on the Miami standards to include things like a publishable study has to have either dose response or time course, and preferably both. So that information really ought to be part of these studies anyway, and so I think that we need to make sure that we've accounted for them in the databases that we're developing. I know I could get arguments from some people in this room, but a lot of us feel that That's useful to know. Thank you. In any case, I think that this is one of those things where a lot of us feel that toxicity is only important in the context of dose. I mean, it's not just an intrinsic property. The second thing that I'd like for us to think about and maybe have people react to is, Chris brought up the idea that everything, he considers everything that his lab produces to be artifact until proven otherwise, and I realize that that's a rhetorical flourish, but I think it's important, and maybe one of the things that we ought to be thinking about as we start to query these databases is that we start thinking about some sort of Miami-like standard for what constitutes just sort of the initial hypothesis generation where one would want to cast a broad net versus the level of rigor in terms of these analyses of metadata of large data sets that would be say of publishable quality. So how much of the artifact do we evaluate and then remove from our final conclusions?
Yeah, I just want to get back to metabolism again. Here, with the ToxCast of looking at issues like prioritizing chemicals for further testing, and of course, if metabolism hasn't been addressed, then that's a huge issue in terms of false negatives. So in moving forward, I realize this is we're just starting out here, and there's a whole issue also of extrahepatic metabolism that also can be very, very important. And oftentimes we're not just talking about one active metabolite, we're talking about a spectrum of metabolites affecting either the same or different pathways. So I think it's, we're early out, and I'm just wondering in terms of a strategy what your thoughts are as we move forward, recognizing these limitations in metabolism and thinking about timeline and what some of the key things are that we want to do now versus later. For example, with ToxCast and some of these early looks to be able to think about where we are now and how ready are we to actually use these in terms of prioritization or early decision making?
So we have used the Solidus assay. There's actually another assay which we believe is giving us information on the hepatic metabolism and it directly tied to some sort of a readout. Another assay that we're actually running right now is the standard one where you simply look at disappearance apparent. So we'll be at least able to say for all the chemicals we look at which ones are metabolized and which ones are not. And it's, I don't remember what the number is, but it's maybe 30% of the chemicals we're looking at are metabolized away, and the other 70% are not. And so those 70% in the current set, okay, you don't have to worry about that so much. The others you obviously do have to worry about because they're getting metabolized away. We're actively searching for partners who have other technologies. Okay, so I think that's really the simple answer is that we don't know. We don't currently have an assay that answers these questions. We're looking to partner with people who have assays to run, and then by combining those assays with all this other data we have, we can help validate both halves of it.
I would also say that there's a time frame. At some point, we'll understand all of metabolism. We're not there yet, but using the rapid throughput approaches, you can just brute force your way through it. In reality, you really could generate every potential breakdown product, metabolite, and ask independently or in combinations, in fact, kinetically, what's the impact in these systems. So you could, rather than focusing it down, make it even bigger and might actually answer some of those metabolism questions if your systems are amenable to that.
Yeah, and so we've kind of thought about that as there are these pieces of software that will predict metabolites. Okay, and so we're actually running all of our parent compounds through those and getting the list of all the metabolites. And then we go out and say, can we actually buy that metabolite? And it's actually the chemical procurement, which is turning out to be the most difficult piece of that. But where you can actually just go buy the metabolite, then we can go and test them.